Welcome to Science with Tal. In today's video, we will conclude our conversation on the fundamental mechanisms of neurons. Before I start, I want to thank each and every one of you for 100 subscribers. I am really glad that my videos have been helpful, and I hope that this discussion will be as well. Now, as a brief reminder, in our last two discussions, we discussed how signals propagate in neurons, and we used the neuromuscular junction to build a foundation to understand signal transmission. In the first full discussion on signal propagation, we mainly covered the passive and active forms of signal conduction in the neuron as modeled by the Hodgkin and Huxley model. To understand these topics, we also had to understand the medium in which neurons are baited that allows this signal to propagate. We also introduced very important neuroscientific techniques such as the voltage and patch clamp. In the second full discussion, we moved on to the neuromuscular junction to see how this simple synapse helps us further understand the interactions between neurons. From this model, we were able to describe the role of calcium in neurotransmitter release, the vesicle cycle, the postsynaptic effects of transmitter release, and the main experiments that allows us to deduce these fundamental mechanisms. Now, to get a full understanding of the signal propagation and the neuromuscular junction as a synapse, I suggest you to watch these videos as they go in depth on each topic they cover. Obviously, as this video builds on the work made by the last two full discussions, there will be some overlapping concepts, but I will make sure that everything you need is also present here. With this being said, let's go back to where we left off. Although the neuromuscular junction is a very appropriate model to describe how chemical synapses function, it still lacks a lot of features that synapses in the central nervous system have, which we are now ready to consider. The first main difference is the quantity of innervation that the postsynaptic cell receives. At the neuromuscular junction, the muscle fiber is usually innervated by one motor neuron, whereas in the central nervous system, neurons can receive up to 1,000 connections. Secondly, the signal transmission in the neuromuscular junction is mediated by a system of one neurotransmitter, acetylcholine, binding to one postsynaptic receptor, the ionotropic nicotinic acetylcholine receptor. As a result of this simplicity, muscle fibers only receive excitatory inputs. On the other hand, chemical transmission in the central nervous system can be mediated by many different neurotransmitters, which bind on many different receptors that can be either ionotropic or metabotropic. Due to this diversity, postsynaptic neurons can receive excitatory or inhibitory inputs. Another very important distinction between the two systems is that action potentials in the neuromuscular junction will usually produce an action potential in the muscle fiber. As we've discussed, the threshold for action potential in the muscle fiber is rather low, so the effects of acetylcholine are very strong. In the central nervous system, the presynaptic inputs are generally weak, such that it takes many excitatory inputs to generate an action potential in the postsynaptic cell. Also, given the addition of inhibitory inputs on the postsynaptic cell, it adds an additional layer of integration that we will have to consider. In all, there are 5 main differences between the two systems that we shall consider in this section. Luckily for us, the work we've made with the neuromuscular junction, such as the vesicle cycle or our understanding of ionotropic channels, is still very valuable and will apply to the central nervous system. With this being said, I want to begin our conversation on neurons in the central nervous system by first describing what metabotropic ion channels are and what purposes they serve. In the brief introduction about chemical synapses, I've introduced metabotropic receptors as receptors that have indirect effects on the cell through biochemical signaling pathways, and that their effects can last considerably longer relative to ionotropic receptors. In terms of what we will cover in this video, we will go over 2 major families of metabotropic receptors: the G-protein coupled receptors, and the receptor tyrosine kinase. By examining these 2 classes of metabotropic receptors, we will be able to understand how the amplification and the modulation occurs in neurons. Let's first cover the G-protein coupled receptors, or simply GPCRs. So, all GPCRs consist of 7 transmembrane alpha helical regions, and as every receptor does, it contains a binding site for a specific ligand, which in our case will be a specific neurotransmitter. As their name entails, these metabotropic receptors are coupled to a G protein. G proteins can come in many different forms and subunits, but for this video, we will consider the most relevant type that applies to neurons, which is the heterotrimeric G protein. Here, heterotrimeric means that the G protein is made out of 3 different subunits: one alpha, one beta, and one gamma. The alpha subunit is loosely associated with the membrane, whereas the beta and gamma subunits form a strong bound complex that is more tightly associated with the membrane. At rest, the alpha subunit is bound to a GDP molecule, and the G protein is said to be in the inactive state. Now, to activate the G protein complex, it requires that the ligand on the given GPCR binds to the binding site of the receptor. When that happens, it changes the conformation of the receptor such that the G protein can now bind to the receptor. This binding promotes the exchange of GDP for GTP. The binding of GTP then causes the alpha subunit to dissociate from the beta-gamma complex. Under this state, the G protein is now considered to be in the active state. Active alpha and beta-gamma subunits can now interact with a variety of effector proteins that either directly influence the behavior of the cell or produce diffusible second messengers that in turn trigger further biochemical cascades. The interaction between the alpha G protein and the effector will also accelerate the rate of GTP hydrolysis such that the G protein can be reformed. To every GPCR pathway, there are always the same four components: the ligand that activates the receptor, the G protein, the receptor itself, and then the effector, which can actually include many effectors, not just one. Now, as long as the ligand is bound to the receptor will remain constitutively active and continue to switch the G protein in the active state. As a result, one receptor can activate many G proteins and produce an amplified signal. It is only when the ligand unbinds that the signal can be turned off. One last thing that I want to emphasize before we examine actual examples of GPCRs is that for each component, ligand, receptor, G protein, and effector, there are hundreds of different types that all have different functions in the cell and lead to different biochemical cascades. Hence, the way I will go about to differentiate all the various mechanisms is by considering what type of G protein interacts with the receptor we are covering. In this video, we will consider three different G protein pathways: GS, GI, and GQ. As I discuss these mechanisms, I will introduce the concept of second messengers and phosphorylation. The first G protein pathway that we will consider is the GS pathway. Here again, we have our metabotropic G protein coupled receptor and our G protein. This pathway begins just as we have previously described. The ligand of the GPCR binds and induces a conformational change in the receptor. The activated receptor can now exchange the GDP from the alpha subunit to a GTP molecule, which now activates the G protein and causes the dissociation of the alpha subunit. The activated alpha S subunit can now interact with its effector protein. The main effector of the alpha S subunit is the integral membrane protein adenylyl cyclase, which becomes stimulated when the alpha S subunit binds to it. This stimulation of adenylyl cyclase leads it to catalyze the conversion of ATP to CAMP. CAMP here is what we refer to as a second messenger. Now, obviously, by saying second messenger, it entails that there is a first messenger, and indeed there is. The first messenger here is the ligand that binds to the receptor, or in other words, the extracellular signaling molecule. Hence, we can define second messengers as intracellular signaling molecules. Second messenger is a fairly common term used in biology, and it covers a vast array of molecules like CAMP in this case, and later we will see other examples like calcium, IP3, and diacylglycerol. Due to the diversity of molecules and their downstream substrates, they have a diverse number of functions ranging from cell proliferation, differentiation, migration, survival, apoptosis, phosphorylation, depolarization, and so on. Anyhow, in this case here, CAMP here acts as a second messenger, and one of its major targets is the CAMP-dependent protein kinase, also known as protein kinase A, or simply pKa. pKa is a dimer of two regulatory and two catalytic subunits. The catalytic subunits are the units of the kinase that phosphorylate other substrates. At rest, when there is no CAMP, the regulatory subunits bind and inhibit the catalytic subunits. As you can see, the regulatory subunits each have two binding sites for CAMP, and upon binding of 4 CAMP, the inhibition is relieved, which frees the catalytic subunits. The free catalytic subunits are now able to phosphorylate substrates at hydroxyl groups of their specific serine and threonine residues. The most common substrates of the catalytic subunits for pKa in neurons include voltage and ligand-gated ion channels, proteins on synaptic vesicles, certain enzymes that are involved in transmitter biosynthesis, and proteins that regulate gene transcription. The phosphorylation by pKa is terminated by a category of enzymes named phosphatases, which cleave the phosphoryl group off of phosphorylated proteins. Now, as a consequence of binding to adenylyl cyclase, the rate of GTP hydrolysis in the alpha S subunit is accelerated to make GDP. This inactivates alpha S and dissociates it from adenylyl cyclase. The free alpha S can reassociate with the beta-gamma complex. Now, remember that as long as the ligand is bound to the GPCR, the receptor can exchange GDPs for GTPs. As such, the receptor can activate many alpha S subunits that can in turn stimulate many adenylyl cyclases. Now, many active adenylyl cyclases also contribute to the amplification of the signal, as while they are active, they catalyze the conversion of multiple CAMPs. Multiple CAMPs can then affect multiple pKas and generate high amounts of phosphorylation because here again, the catalytic subunits can phosphorylate many compounds while they're active. In the GS example, the even steps 2, 4, and 6 correspond to amplification steps as shown by the yellow circle. Remember that the main feature GPCRs provide is the amplification of the signal, and it is very well illustrated here. Back to the schematic, here is a condensed view of what the GS pathway provides, which essentially summarizes what we have just discussed. As a side note, I want to mention that the S in the alpha S subunit is conveniently chosen to be S because the subunit stimulates adenylyl cyclase. This pathway is in direct opposition with the alpha I pathway, which as you can see stands for inhibitory. As you can see, the alpha I pathway is essentially the same in its shape, but the result from alpha I binding to adenylyl cyclase is inhibition. As a result, the levels of CAMP, active pKa, and overall phosphorylation are decreased. Now that we have covered the GS and GI pathways, let's consider the GQ pathway. Just like the other GPCR pathways we have covered, the mechanisms begins with the ligand of the GPCR binding to it and causing a conformational change to the receptor. The G protein binds to the receptor, and that causes the subsequent exchange of GDP for GTP. Here again, this exchange causes the dissociation of the alpha Q and beta gamma subunits. The active alpha Q then stimulates phospholipase C, also known as PLC, which is a particular enzyme with the main role of hydrolyzing substrates. When active, the substrate that PLC hydrolyzes is phosphatidylinositol 4,5-biphosphate, or more simply known as PIP2. When we take a look at the molecular structure of PIP2, you can see that there are two main components. First, there is a ring with 3 phosphate groups that is called inositol 1,4,5-triphosphate or IP3, and secondly, there are 2 fatty acid chains that form diacylglycerol or simply DAG. The hydrolysis of PIP2 leads to the production of 2 second messengers, DAG and IP3. Now, as you can see, DAG is hydrophobic and remains in the membrane. The formation of DAG will lead to the recruitment of protein kinase C or PKC. PKC is a monomeric kinase that has a catalytic and regulatory subunit. To remove the inhibition from the regulatory subunit, the kinase binds with DAG at the membrane. Then, just like pKa, PKC phosphorylates substrates to generate cellular responses, and then the phosphorylation is removed by phosphatases. Now, the other substrate made from PIP2 is IP3. In comparison to DAG, IP3 is diffusible in the cytosol. One of its important downstream functions is being a ligand for the IP3 receptors that are located on the membrane of the smooth endoplasmic reticulum. The smooth endoplasmic reticulum in cells is usually known to be a site for lipid synthesis, but one other important function is being a storage site for calcium. Since there is way more calcium inside the smooth endoplasmic reticulum, it turns out that upon opening, IP3 receptors let calcium flow out into the cytosol. Now that there is calcium in the cytosol, It gets very interesting because calcium has a lot of functions as a second messenger. In previous discussions, we've talked about calcium as being the signal for transmitter release, but it turns out that calcium has many more roles as a second messenger. For example, calcium can bind to the ryanodine receptors that are also on the smooth endoplasmic reticulum, and this leads to the opening of calcium-gated channels in the membrane, which lets even more calcium flow into the cytosol. Moreover, calcium can bind to the small cytoplasmic protein named calmodulin. The calcium-calmodulin complex then activates the calcium-calmodulin-dependent kinase, also commonly referred to as CaMK. Like the other kinases we've discussed, CaMK goes on to phosphorylate other substrates. Again, these substrates can include ion channels, vesicle proteins, transcription factors, and so on and so forth. Now, as a small tangent, I want to touch briefly on the structure of one particular subtype of CaMK kinase, which is the type 2 or simply CaMK2, because it has a particular property that differentiates it from other kinases. So, similar to other kinases, CaMK2 has catalytic subunits that are inhibited by regulatory domains at rest. To remove this inhibition, it requires binding of the calcium-calmodulin complex. Now, here's where it gets interesting. Once it's activated, CaMK2 can phosphorylate itself by a process of autophosphorylation, which can make the enzyme persistently active even when the calcium is not there anymore. This kinase is very important because it has important implications for neuronal plasticity, which is a concept we'll discuss later. Alright, now that we've covered the main events of the GQ pathway, here is what it looks like as a whole. As a small detail, I want to mention that calcium heavily contributes to the activation of PKC, so this is a noteworthy detail to consider. Now, the pathway is very complex, so make sure to go slowly through it to understand, and if there's any issues with it, make sure to let me know. Back to the schematic, we can simply summarize the GQ pathway in this way. The activation of alpha Q leads to the activation of PLC, then formation of two second messengers, DAG and IP3, and then the activation of kinases that increase the phosphorylation inside the cell and the activation of calcium-dependent proteins. Alright, we've now covered what I generally wanted to discuss when it comes to GPCRs. Obviously, there are a lot of details to add on to what I showed, which I will do in due time, but for the moment, this should suffice us to get a good grasp of what is the function of GPCRs. Let's now cover the tyrosine kinase receptors, which are the second main class of metabotropic receptors. Let's start with the structure of these receptors, which are often simply abbreviated to RTKs. RTKs are rather different from G-protein coupled receptors, as they have only one subunit with an extracellular binding domain. In the intracellular portion of the receptor, it has a kinase domain, and the two domains are connected via a transmembrane alpha helix. As we've learned in the previous section, kinases are a particular set of enzymes that take one of the three phosphate groups of ATP and transfer it on a given substrate. This process is referred to as phosphorylation, and it usually leads to the activation or deactivation of the substrate. Examples of substrates can be ion channels, vesicle proteins, transcription factors, and many more. The phosphorylation is then removed by special proteins named phosphatases. I want to mention as well that this phosphorylation reaction often occurs on tyrosine residues, and the receptor itself has a lot of these residues, which explains why it is called the receptor tyrosine kinase. Now, due to the fact that it has these tyrosine residues, the receptor can phosphorylate itself along with other proteins, but we will get back to that aspect shortly. As mentioned earlier, the receptor has a ligand binding site. In contrast to GPCRs that are commonly activated by typical neurotransmitters like dopamine, glutamate, acetylcholine, and so on, RTKs are commonly activated by peptide hormones such as epidermal growth factor, fibroblast growth factor, nerve growth factor, and brain-derived neurotrophic factor. Now, let's see how these receptors get activated. At rest, when the RTK has no ligand bound to it, a good majority of RTKs exist as monomers. When two separate monomeric receptors bind to their respective ligands, it causes dimerization between the two. The formation of the dimer is very important because it will lead each respective RTK to phosphorylate the other RTK until every tyrosine residue has a phosphate group. At this stage, we say that the dimer is fully phosphorylated and fully activated. Now that the RTK is active, it will act as a kinase to other substrates and activate multiple downstream pathways. As you can imagine, since it's a kinase, there's an enormous quantity of possible downstream pathways. One notable pathway that we can consider is actually one that we have covered already, and it is the PIP2-PLC pathway. This is the GQ pathway of GPCRs that I have covered previously, and I suggest you to watch the section on this pathway in the GPCR portion to get more information and more detail about it, but it never hurts to briefly go over this pathway again. So, the main difference simply lies in what activates PLC. In this case, after the RTK dimerizes and becomes an active kinase, it activates PLC, which leads to the subsequent hydrolysis of PIP2 into IP3 and DAG. On one end, DAG leads to the activation of PKC, which is a kinase that then goes on to phosphorylate some substrates in its respective downstream pathways. IP3 leads to the increase of intracellular calcium by opening calcium-gated channels in the smooth endoplasmic reticulum. The entry of calcium can either open more calcium channels or it can bind to a protein named calmodulin to activate another kinase named CaMK. The activation of PKC and CaMK then leads to an increase in phosphorylation that can activate multiple downstream substrates like ion channels, transcription factors, and so on. This is good to know, but it would be obviously more enlightening to cover a pathway that is more a hallmark of the receptor tyrosine kinase. This pathway in question that is famously associated with RTKs is the MAPK pathway. To understand the logic of this pathway, let me first start by describing what MAPK does, and then we'll see how the RTKs relate to it. So first off, MAPK stands for mitogen activated protein kinase. As it says in its name, MAPK is a kinase, and its main phosphorylation targets are involved in gene transcription, cell growth, cell differentiation, to name a few. To contribute to these functions, MAPK is able to translocate to the nucleus and phosphorylate different proteins such as transcription factors that regulate the formation of other proteins that are involved in the cell cycle and cell differentiation. One important aspect about MAPK that is different from other kinases that I've covered like pKa, PKC, and CaMK is that to activate MAPK, it requires phosphorylation. Remember that to activate the other kinases, it required the removal of inhibition by regulatory subunits by binding certain molecules to the kinase. Here, it simply requires that MAPK gets phosphorylated. Alright, now that we have an intuitive idea of what MAPK does, let's see how it gets activated. So, after dimerization and activation of RTK, a protein by the name of GRB2 comes and binds to a phosphorylated tyrosine. To bind to the phosphorylated tyrosine, GRB2 has a particular domain named SH2 that has the particular sequence that allows it to bind to phosphorylated tyrosines. GRB2 also has two additional domains named SH3 that bind to proline-rich sequences in another protein named SOS. As you can see, GRB2 acts as an adapter protein to link the activation of the RTK to SOS. Now, SOS will then activate a G protein by the name of RAS by exchanging GDP for GTP. In the biology jargon, we refer to SOS as a guanine nucleotide exchange factor, or GEF. Now, RAS is a monomeric G protein. Remember that in our discussion on GPCRs, we covered a heterotrimeric G protein composed out of an alpha, beta, and gamma subunit. Here, RAS is only one subunit, but it nonetheless functions in the same way as the trimeric one. When it is bound to GDP, it is inactive, and when it is bound to GTP, it is active. From this activation, you can notice a rather important difference between RTKs and GPCRs because RTKs do not activate the G protein directly. Indeed, it is the activation of multiple adapter proteins that leads to the switch between GDP and GTP in the G protein, whereas GPCRs directly made the G protein active. Alright, now that RAS is active, RAS removes autoinhibition in another kinase called RAF, but this does not fully activate RAF just yet. To fully activate RAF, RAF must dissociate from RAS, and the dissociation is accompanied by the hydrolysis of GTP to GDP. It turns out that RAS G proteins are rather slow at hydrolyzing their GTP. For that reason, additional proteins called GTPase activating proteins, also known as GAPs, like RAS-GAP, bind to the G protein to facilitate the hydrolysis reaction. If you recall our discussion on GPCRs, this constitutes a rather important difference again with trimeric G proteins, because alpha subunits were able to hydrolyze their GTP to GDP by themselves after interacting with their effector proteins. Here, RAS cannot do this hydrolysis quickly on its own, and that is why it needs an additional protein to terminate the signal. Alright, now that RAF has dissociated from RAS and is active, it contributes to phosphorylating another kinase named MEK. MEK then links our pathway to MAPK by phosphorylating it and thus activating MAPK. As mentioned, MAPK then phosphorylates and contributes to many downstream signals that are involved in cell growth, differentiation, proliferation, and so on, and their actions often end up being on transcription factors. Now, before we move on, I want to point out a few details on this pathway because this illustration is not fully accurate, so for the sake of correctness, let me point out a few things. First, it is noteworthy to mention that RAS is generally membrane-bound. Secondly, the autoinhibition in RAF is carried out by a small molecule named 1433, and the binding to RAS causes the dephosphorylation of 1433 and the partial activation of RAF. Thirdly, when RAF is activated, it forms a dimer. But, these are small details that you can consider on your own, but in all, the most important points from the MAPK pathway are shown here. Alright, now that we have covered the two main categories of metabotropic receptors, let's continue discussing the differences between neurons at the neuromuscular junction and neurons in the central nervous system. The next topic I want to cover will actually cover points 2, 3, and 4 altogether, which are that inputs can be excitatory or inhibitory, that the postsynaptic receptors can be ionotropic or metabotropic, and that the presynaptic cells can have different types of neurotransmitters. To cover these points, we will explore the main neurotransmitters as well as their receptors in the central nervous system. For the moment, the only neurotransmitter that we have covered in great detail is acetylcholine, which is the main excitatory neurotransmitter in the peripheral nervous system. In the neuromuscular junction portion of our discussion, I went thoroughly over the synthesis, the packaging, the release, and the termination of acetylcholine in these two videos titled The Vesicle Cycle Explained and The Neuromuscular Junction Synapse Explained. In these videos, I've also explained in great detail the main ionotropic receptor of acetylcholine, which is the nicotinic acetylcholine receptor. Now, for the moment, we've only talked about acetylcholine in the peripheral nervous system, but it turns out that acetylcholine is also important in the central nervous system. Acetylcholine effects in the central nervous system are mostly mediated through a metabotropic receptor, and that receptor is called the muscarinic acetylcholine receptor. So, to complete our picture on acetylcholine, let me explain how this receptor functions. To do so, let's take a look at a cholinergic synapse in the autonomic ganglia of the peripheral nervous system. The synapse presynaptically releases acetylcholine, and the postsynaptic cells contains nicotinic acetylcholine receptors, as well as the muscarinic acetylcholine receptors. First of all, just like I've explained for the nicotinic acetylcholine receptor, the muscarinic acetylcholine receptor is called that way because muscarine, a chemical made from certain mushrooms, can bind to the receptor and open it. For that reason, muscarine is referred to as an agonist. This receptor also has antagonists, such as scopolamine, atropine, and ipratropium, that are able to bind to the receptor but not activate it. Now, when the synapse is fired and acetylcholine is released in the cleft, the binding of acetylcholine to nicotinic acetylcholine receptors will cause a fast EPSP, or in other words, a fast depolarization. Additionally, acetylcholine will bind to the metabotropic receptor, which will cause a depolarization but in a bit of a different way. Let me explain how. If we take a closer look at the postsynaptic terminal where the muscarinic receptor is, it turns out that the postsynaptic side also has some potassium channels that are there. Now, two things to note that are important to understand how the receptor mediates the excitation of the postsynaptic cell is that first, the muscarinic receptor in this case mediates a GQ-type GPCR. To refresh your memories, remember that this is the basic GQ pathway where the activation of the GQG protein leads to the activation of PLC, which hydrolyzes PIP2 into DAG and IP3. DAG and IP3 are two second messengers, and they lead to the activation of downstream kinases. If you need more information on this pathway, consider watching the section on GPCRs where I thoroughly go over it. Alright, now the second thing you need to know is that these potassium channels bind PIP2 as a cofactor to stay open. Therefore, when the muscarinic receptors get active, they activate GQG proteins that in turn activate PLC. PLC hydrolyzes PIP2 into IP3, which in turn closes the potassium channel because there is no more PIP2. As a consequence of potassium channel closure, it makes the postsynaptic cell more excitable. Remember that potassium currents are outward, so in other words, hyperpolarizing. Hence, removing these currents removes currents that counter the depolarization from nicotinic acetylcholine receptors, and more importantly, the reduction of potassium currents lowers the threshold to fire and action potential. Also, due to the fact that this is mediated by a GPCR, the effects last considerably longer than the quick depolarizations from the anotropic channels. Hence, in this case, the muscarinic receptor mediates a slow and sustained depolarization. Now, you need to know that GPCRs can have different functions at different places, whereas ionotropic receptors will most often have the same effects regardless of where they are expressed. For example, nicotinic receptors will mediate a non-selective cation current, regardless of if it is at the neuromuscular junction or here at the autonomic ganglia. However, the same cannot be said for GPCRs like the muscarinic receptor. To give you another example of what the muscarinic receptor can do, let's consider the synapse between the vagus nerve and the heart. For this synapse, let's simply consider the actions of the muscarinic receptor. In this synapse, there are also potassium channels that are expressed on the postsynaptic side. Here, when this synapse fires and releases acetylcholine that binds onto the muscarinic receptors, it causes a hyperpolarization by opening potassium channels. To understand what's going on, let's take a look at the muscarinic receptor. It turns out that in this case, the G protein that the muscarinic receptor activates is a GI type. As a reminder, the GI pathway leads to these downstream signals, but it's not that important for us because it turns out that this hyperpolarization here is not even mediated by second messengers. When acetylcholine activates the muscarinic receptor, the receptor activates the G protein, which dissociates the beta, gamma, and alpha subunits. The active alpha subunit goes on to interact with other downstream signals, but it turns out that it is the beta and gamma subunit that goes on to interact with the potassium channel to open them. The opening of these channels hyperpolarizes the cell towards the equilibrium potential of potassium, which is always lower than the resting membrane potential. In terms of the heart, the activation of the vagus nerve and the muscarinic receptors causes the heart to beat slower. Also for reference, these potassium channels that the beta-gamma subunit open are named G-protein-coupled inwardly rectifying potassium channels, or simply GERC. With this covered, we now have a good idea behind what's going on with acetylcholine across the body. The next neurotransmitter that I want to cover is one we haven't seen yet, and it is glutamate. Glutamate is the major excitatory neurotransmitter in the brain and the spinal cord. To understand its function, let's consider a synapse with presynaptic glutamate and the respective postsynaptic receptors. Let's first start presynaptically and discuss the life cycle of glutamate from the synthesis to its recycling. Glutamate is directly synthesized at the terminal from glutamine in a straightforward reaction catalyzed by the enzyme glutaminase. To get the transmitter inside the vesicles have a V-class ATPase that pumps protons inside the vesicle, and then the proton gradient is used to import the glutamate inside the vesicle. The transporter that mediates this exchange is named the vesicular glutamate transporter. Now that the vesicles are full of glutamate, they can be released in the cleft by the same general principles that we've covered in the vesicle cycle section. The action potential depolarizes the cell, opens voltage-gated calcium channels, which fuses the vesicles and releases glutamate in the cleft. When glutamate is in the cleft, it will be able to bind to its various receptors. But before I move on to the specific receptors of glutamate, I want to first discuss how the signal is terminated because it uses a different mechanism that we haven't seen yet. In our discussion on acetylcholine, remember that the mechanism to terminate the acetylcholine signal is to degrade it through a specific enzyme called acetylcholine esterase, which turns acetylcholine into acetate and choline. This type of mechanism is generally labeled as neurotransmitter degradation. In the case of glutamate, the mechanism that terminates the signal is neurotransmitter reuptake. An integral component of this mechanism is a glial cell named the astrocyte. Astrocytes participate both with the presynaptic and the postsynaptic cell, and for that reason, it is often said that the three structures form a tripartite synapse. Now, when glutamate is in the synaptic cleft, it can enter the astrocyte and the presynaptic cell through a class of transporters called the excitatory amino acid transporters, or simply EAAT. These transporters co-transport glutamate with sodium. The glutamate that is transported into the astrocyte is converted back to glutamine by the enzyme glutamine synthetase. This glutamine is then transported out of the astrocyte by a different transporter named the System N Transporter 1 or SN1, which co-transports sodium as well. The glutamine that has been extruded from the astrocyte can then enter the presynaptic terminal through another transporter called the System A Transporter or SAT2 and be reconverted into glutamate by glutaminase. This final step ties in the reuptake process of glutamate. I want to mention as a side note that the glutamate can also be synthesized at the presynaptic terminal from glucose. As glucose gets oxidized in the mitochondria through the citric acid cycle, it eventually forms a compound by the name of alpha-ketoglutarate. Alpha-ketoglutarate can get converted into glutamate by an enzyme called glutamate dehydrogenase. Last note, I want to mention that you might find in some literature that the excitatory amino acid transporters or EAAT are also named GLAST and GLT, which stand for glutamate aspartate transporter and glutamate transporter. Back to the synaptic cleft, let's now see what receptors glutamate interacts with. These receptors can either be ionotropic or metabotropic. So let's start with the ionotropic receptors first. There are three major types of ionotropic receptors: NMDA, AMPA, and kainate. Similar to acetylcholine receptors, the nomenclature for these receptors comes from the agonists that open them. These three receptors also have antagonists that block their opening. The drug AP5 blocks the NMDA receptors, and the drug CNQX blocks the AMPA and kainate receptors. These compounds will be important to consider later as they will help us determine some properties of the channels. Alright, now when glutamate does bind and opens each channel, the three mediate an inward current that depolarizes the cell when the membrane potential is negative. The inward current from the three channels is composed out of a potassium and sodium component, just like the nicotinic acetylcholine receptor. One important detail is that the NMDA receptor, contrarily to the AMPA and the kainate receptor, is highly permeable to calcium, but this is a detail I'll come back to shortly. When we consider the IV curve for these receptors, the reversal potential for each is near zero, so about in between the reversal potential of sodium and potassium. If you want to get more information on IV curves and how we can determine which ions go through ligand-gated receptors, I suggest you to consider going to the neuromuscular junction video where I thoroughly explain how IV curves can be made for acetylcholine receptors from patch clamp results. Since glutamate receptors, especially AMPA and kainate, behave in the same way as the nicotinic acetylcholine receptor, the video should be very helpful if needed. Now, in terms of structure, the three receptors have all the common features that we have discussed in previous sections about channels. They have an extracellular ligand binding region and a pore that allows the flow of ions. One thing about the structure of these channels that I want to mention though is that the specificity to calcium for the NMDA receptor comes from the fact that it has an important asparagine residue in its pore that the other channels don't have. Speaking of the NMDA receptor, I want to analyze it a bit further because it is a rather unique receptor among the ligand-gated channels in its ability to conduct current. For the NMDA receptor to open, it requires two factors. First, the receptor must bind its ligand, which, as we've discussed, is glutamate, but it turns out that to open the NMDA receptor, it also requires the binding of glycine. Glycine is a neurotransmitter that we will cover later that is involved in some inhibitory circuits, but here it acts as a co-agonist to the NMDA receptor. Now, to understand the second factor, you must know that at rest, magnesium binds tightly to the NMDA receptor and essentially acts as a plug to the channel. This magnesium interaction blocks any synaptic current from occurring, even if glutamate and glycine are bound to the receptor. To remove magnesium from the pore of the channel, it requires a sufficient depolarization that will kick it out. Indeed, given that magnesium is a positively charged ion, it is attracted to the negative membrane potential of the neuron, and that is why it blocks the pore, but as the membrane potential gets depolarized, let's say from AMPA activity, the membrane potential becomes progressively more positive and now repels the magnesium out of the NMDA receptor. When the magnesium is removed, the ions can flow and the NMDA receptor produces an excitatory current. Because the NMDA receptor requires the binding of neurotransmitters, which come from the presynaptic side, and also a depolarization to kick out magnesium that comes from the postsynaptic side, the NMDA receptor is often referred to as a coincidence detector. In this case, the coincidence comes from the dual activity in the pre- and postsynaptic sides. One question to ask ourselves now is what exactly is the purpose behind the dual gating of the NMDA receptor? One good insight to this question is to remember that the receptor conducts calcium. As we've discussed in our conversation about G-protein coupled receptors, calcium can be used in the body as a second messenger to activate kinases and so on. For example, in the GQ pathway, calcium can bind to calmodulin and activate calcium-calmodulin-dependent kinase. The kinases will then go on and interact with downstream substrates that might lead to the formation of new proteins. Hence, the entry of calcium can have very important long-lasting effects on the neuron beyond simply depolarizing it. As a quick foreshadowing note, NMDA receptors are very important in mechanisms that underlie synaptic plasticity due to their high calcium permeability, but we will come back to that aspect in due time when we discuss plasticity. Now, this removal of magnesium essentially constitutes a form of voltage dependence for the NMDA receptor, which is perfectly illustrated in its IV curve. As you can see, at negative membrane potentials when the magnesium blocks the receptor, there is no current because ions cannot flow. But when the membrane potential reaches a certain point, the current from the NMDA receptor becomes inward, which shows that the magnesium has left. As you go more and more positive, the IV curve of the NMDA receptor morphs with the IV curve of the non-NMDA receptors because obviously there is no more magnesium to block the channel. Beyond the different IV curves, NMDA receptors and the other glutamate receptors also differ on the time course of their current. To see this relationship, let's consider a plot of currents as a function of time. To obtain the results from all these receptors, we will perform a voltage clamp experiment on a terminal that contains these receptors. As a brief refresher of the voltage clamp technique, remember that the purpose of this method is to maintain the membrane potential of the neuron at a fixed voltage and measure what current it takes to maintain it at that voltage. Hence, the current you will see in the graph corresponds to the compensatory current. The voltage clamp is explained in another video on the channel, so make sure to watch it if the method is still unclear. Now, one caveat with these receptors is that how can we tell what current goes through the NMDA receptor if the AMPA receptor or kainate receptor opens as well? One tool that we can use to isolate the current is by using some pharmacological blockers that prevent synaptic activity in the channel we do not want to measure. The pharmacological blockers in question are the antagonists I've pointed out. Hence, by considering a postsynaptic terminal with NMDA and AMPA receptors only, let's start by administering AP5 to the cell. As AP5 blocks the NMDA receptors, the voltage clamp results for the AMPA receptors are the following. To see how they compare to the NMDA receptors, we can perform the voltage clamp, but we now use CNQX to block AMPA receptors. The results for the NMDA receptors are the following. As you can see, when NMDA receptors open, the current rises and decays with a much slower time course than the AMPA receptors, which rise and decay very rapidly. When it comes to kainate receptors, they generate a current that rises quickly but decays more slowly than AMPA receptors. You can imagine that to isolate their current, it requires a similar experimental setup as what I explained for the NMDA and AMPA currents. Alright, now that we understand the basics of glutamate synthesis, reuptake, and its receptors, I want to drive home a final concept that is very important in neurons of the central nervous system. As you can imagine, it is very important that the reuptake process of glutamate functions properly and efficiently to remove any extra glutamate that might be diffusing in a cleft. Indeed, if that glutamate is not removed properly, the glutamate might keep binding to the receptors and this will cause them to get constantly active. This overstimulation by glutamate is often referred to as glutamate excitotoxicity, and excitotoxicity can often lead to seizures or other abnormal firing patterns. Also, excitotoxicity can pose a particular problem with NMDA receptors because they are highly permeable to calcium. Thus, if the internal calcium concentration rises too much because the NMDA receptors are overactivated, it can lead to cell death because high calcium concentration activate proteins that end up killing the cell. Now, to finish our discussion on glutamate and its receptors, I want to briefly touch on the metabotropic receptors of glutamate. I will be a bit brief with these receptors because I have covered a lot of information on their mechanism of action already in our more general discussion on GPCRs. Nonetheless, I want to still give a few details about these receptors so we have a good and intuitive idea of what they do. First thing, metabotropic glutamate receptors can be generally divided into 3 large groups that contain up to 8 different forms of the receptor. These groups separate the mGlu-R based on the similarity of their amino acid composition, the G protein they activate, and what ligands they can bind. What I'll focus on here is the typical G protein they activate and where are they usually located. Note that what I'll say is not to be taken as an absolute because there are obviously exceptions to all these criteria, but they will be good enough for us to get an idea. Alright, so when it comes to group 1, they generally activate the GQG protein, which, as you might remember, activates phospholipase C. This activation leads to the hydrolysis of PIP2 into the formation of 2-second messengers, IP3 and DAG, which subsequently activate different kinases. If you want more information on this pathway, I've thoroughly covered in the GPCR section. The receptors within group 1 are usually located postsynaptically, and their activation usually mediates cell depolarization and increase in neuronal excitability. Additionally, in comparison to ionotropic receptors, which are usually located in direct opposition of the glutamatergic presynaptic terminal, postsynaptic metabotropic glutamate receptors are usually located on the periphery. In comparison to group 1 mGlu-Rs, group 2 and group 3 mGlu-Rs are usually coupled to the GIG protein, which is responsible for inhibiting adenylyl cyclase and the subsequent production of CAMP. As a consequence of this, phosphorylation in the cell is reduced. Group 2 and 3 receptors are also associated with direct regulation of ion channels by the liberation of the beta and gamma subunits. Receptors in group 2 and 3 are often localized presynaptically and generally mediate inhibition of transmitter release. As I said, there are a lot of other mechanisms and pathways that these receptors mediate, but this should suffice for our purposes. Alright, with glutamate now covered, we can move on to the next neurotransmitter. If you recall from the start of this section, one huge difference between neurons in the central nervous system and neurons at the neuromuscular junction is the fact that neurons in the central nervous system can have an excitatory or inhibitory output on the postsynaptic cell. Accordingly, given that we've established that glutamate is the main excitatory neurotransmitter, we need to now consider what is the main inhibitory neurotransmitter. The main inhibitory neurotransmitter in the central nervous system is gamma-aminobutyric acid, or simply GABA. To understand this neurotransmitter, let's first consider its synthesis and signal termination mechanism, and then we'll discuss the postsynaptic receptors as well as what inhibition it provides to the nervous system. Alright, so just like the other neurotransmitters we've covered, GABA is synthesized and packaged at the presynaptic terminal. Interestingly enough, it turns out that GABA is synthesized from glutamate, and this reaction is catalyzed by an enzyme named glutamic acid decarboxylase, or simply GAD. As a noteworthy fact, for GAD to properly function, it requires a cofactor named pyridoxal phosphate, which is derived from vitamin B6. Hence, a deficiency in this vitamin could lead to problems with GABA communications in the nervous system. Now, since GABA is made from glutamate, you will notice that there will be a lot of overlaps between the synthesis and the reuptake process of GABA in comparison to glutamate. Nonetheless, let's continue with the mechanism. After synthesis, GABA is transported into the synaptic vesicles via the vesicular inhibitory amino acid transporter, or VGAT. VGAT uses the gradient of protons that is generated by VATPase to import GABA inside the vesicles. After action potential propagation and calcium-mediated exocytosis of the vesicles, the GABA molecules are released in the cleft and interact with postsynaptic receptors. To remove GABA from the synaptic cleft, inhibitory synapses also form tripartite synapses with astrocytes. Astrocytes and presynaptic terminals have particular GABA transporters named GAT that co-transport GABA with sodium inside. The GABA that directly returns to the presynaptic terminal can get recycled or broken down if it is not needed. In the astrocyte, GABA gets reconverted into glutamate by GAD, the same enzyme that did the opposite reaction in the presynaptic terminal. This glutamate is then converted into glutamine by glutamine synthetase. The glutamine can then leave the astrocyte by the system N transporter and return into the cell through the system A transporter. The two transporters co-transport sodium with glutamine. When glutamine is inside the presynaptic terminal, it gets reconverted into glutamate by glutaminase, which completes our reuptake cycle. When GABA is in the synaptic cleft, it can interact with both ionotropic and metabotropic receptors. The ionotropic receptors for GABA is named GABA-A, and the metabotropic receptor is named GABA-B. Let's start with the ionotropic channel. First, I want to briefly discuss the structure of the GABA-A channel because it turns out that it has a very similar structure to the nicotinic acetylcholine receptor. Indeed, the GABA-A channel is a pentamer, usually composed out of 2 alpha, 2 beta, and 1 gamma or delta subunit. To open GABA-A channels, there are 2 binding sites in between the 2 alpha and beta subunits that upon GABA binding opens the pore. When the pore opens, the receptor conducts chloride inside the cell, but we'll come back to that aspect shortly. Another important structural aspect of this channel is that, in comparison to the nicotinic acetylcholine receptor, which had negatively charged acidic residues near the pore, the GABA-A receptor has positively charged basic residues, which makes the channel selective for anions like chloride instead of cations. Now, in terms of agonists and antagonists, GABA-A channels are highly targeted by several types of drugs. Some notable drugs include benzodiazepines, barbiturates, and alcohol. All these different drugs and GABA bind to different sites on the receptor, but all of them cause the receptor to open or at least increase the likelihood of being open. For example, it is known that benzodiazepines bind between the alpha and gamma subunits, yet, on their own they do not activate the channel completely. However, when GABA is bound and benzodiazepines are also bound, the activity of the channel is considerably increased, which considerably enhances the effects of the GABA-A receptor. Alright, now in terms of its function, the GABA-A receptor is essentially a ligand-gated chloride channel. This means that when 2 molecules of GABA bind, the receptor opens a path for chloride ions to flow. If you recall from some of our first discussions, the respective concentrations in and out of the cell for chloride are about 4 and 110 millimolars. This imbalance in concentrations already hints at the fact that chloride will be driven to enter the cell purely from its chemical gradient. From the Nernst equation, we can compute that the equilibrium potential of chloride is about negative 87 millivolts. Recall that the equilibrium potential is the value at which the chemical and electrical gradients of chloride are balanced such that there is no net current going across the membrane. To visualize the equilibrium potential value better, we can plot the IV curve of chloride. From the conventions that we have established, remember that positive current is called outward and is hyperpolarizing, whereas negative current is called inward and represents depolarizations. Hence, at resting voltages above negative 87 millivolts, the produced current is hyperpolarizing, and below negative 87 millivolts, the current is depolarizing. To see the effects of GABA-A receptors on postsynaptic partners, let's consider a plot of the membrane potential as a function of time for a neuron that has a resting membrane potential of negative 70 millivolts. When GABA-A receptors open, the entry of chloride into the cells pushes the membrane potential towards the equilibrium potential of chloride. Because this hyperpolarization stems from a synaptic event, it is also often referred to as an inhibitory postsynaptic potential or simply IPSP, which directly opposes excitatory postsynaptic potentials or EPSPs that we've previously covered with glutamate and acetylcholine. When this synaptic event happens alone and the equilibrium potential of chloride is below the resting membrane potential, this is considered to be the typical form of inhibition. But a different form of inhibition can occur under different conditions. This form of inhibition is named shunting inhibition, and to understand it, let's consider the plot for the membrane potential as a function of time. In cases where shunting inhibition occurs, there are generally two features that cause it to happen. First, the equilibrium potential for chloride closely equals the resting membrane potential. So, for the purposes of this example, imagine that the equilibrium potential for chloride now equals negative 70 millivolts. The second aspect that is present is an excitatory input, and here, for this example, we will assume that it comes from a glutamate receptor, AMPA. Remember that the equilibrium potential of the AMPA receptor is near zero because the channel is a non-selective cation channel for sodium and potassium. Now, if the GABA channels were to open alone, there would be no net current coming from them because the cell is already at the equilibrium potential for these channels. Inversely, if the AMPA channels were to open alone, their activity would increase the membrane potential towards their own equilibrium potential to reach the threshold. Now, here's where it gets interesting. If both channels open simultaneously, the excitatory input by the AMPA receptor will be lessened in comparison to when it opens alone because the GABA-A channels essentially shunt the depolarization towards the resting state. Indeed, when the two channels open, you can almost imagine their combined activity as a tug of war, and in this instance, the GABA channels pull the AMPA activity down. Another way of conceptualizing shunting inhibition is to think about GABA receptors in terms of positive charges. Indeed, in terms of depolarizations and hyperpolarizations, the activity of positive and negative charges are a bit interchangeable if we inverse their directions. Hence, instead of thinking about negative ions entering the membrane, we can imagine the GABA channels as channels that let positive ions flow out of the cell. Now, due to the fact that currents must always flow in closed loops, you can imagine that when the two channels are activated together, the GABA channels offer an additional path for the positive charges that have entered from AMPA to leave. Again, as a result, the depolarization is shunted. Alright, as a final comment about ionotropic GABA receptors, I want to mention that although GABA is usually covered as an inhibitory neurotransmitter, we are assuming that we are talking about a mature neuron because in the developing brain, GABA forms excitatory connections. To understand how this can occur, remember that whether a channel produces inhibition or excitation is purely a product of the ionic concentrations in which the neuron is baited in. Early in development, the chloride concentrations are mainly controlled by the NKCC cotransporter, which transports 1 sodium, 1 potassium, and 2 chloride ions inside the cell. As a result, the internal concentration of chloride is very high and its equilibrium potential becomes higher than the resting membrane potential. Thus, when GABA receptors open, chloride ions leave the cell and cause a depolarization. In mature neurons, however, the NKCC cotransporter gets downregulated and a new transporter by the name of KCC2 gets expressed. KCC2 exports 1 potassium and 1 chloride out of the cell, which makes the internal chloride concentration very small. As a result, the equilibrium potential for chloride becomes more negative and the IV curve shifts below the resting membrane potential. Now, when the GABA receptors open, it causes a hyperpolarizing current due to chloride efflux. For that reason, you can see that inhibition and excitation comes from the reversal potential of the ions that flow through the channels and not necessarily from the structural properties of the channels themselves. Now that we have a good idea of what is going on with ionotropic channels, let's discuss the metabotropic GABAb receptors. These receptors are generally associated with two main mechanisms of action, although I am more than certain they mediate many more. Nonetheless, let's cover the two that are the most common. The first mechanism from GABAb channels is actually very similar to one we've covered for the muscarinic acetylcholine receptors and the metabotropic glutamate receptors. This mechanism acts through the GIG protein and mediates the activation of GERK potassium channels to inhibit the postsynaptic cell. Indeed, given that the reversal potential of potassium, which is about negative 80 millivolts, is always below the resting membrane potential, the opening of these channels causes an efflux of potassium that hyperpolarizes the cell. The activation of these channels comes from the dissociation of the beta and GABA subunit during GIG protein activation. The second mechanism occurs on the presynaptic side, and here again, the GIG protein is used. When the G protein gets activated, the dissociation of the beta and gamma subunit from the alpha causes the blockage of presynaptic voltage-gated calcium channels. Given that the calcium channels are essential for transmitter release, the blockage of these channels prevents neurotransmitters from being released and signaling onto the postsynaptic cell. Hence, this presynaptic blockage leads to a postsynaptic inhibition. Now, based on what we've discussed on the ionotropic and metabotropic channels, we can establish that there is three general ways to inhibit the postsynaptic cell. The first is through the action of the GABA-A receptor, which hyperpolarizes the cell by letting chloride ions enter. This form of inhibition includes the shunting inhibition we discussed as well. The second way to inhibit the postsynaptic cells is through the activation of postsynaptic GERK channels by GABAb activation, and the third way is by preventing or at least diminishing presynaptic release by blocking presynaptic calcium channels, again by GABAb activation. Before I conclude my discussion on inhibition, I want to briefly mention that there is another important inhibitory neurotransmitter by the name of glycine. Beyond being an important cofactor for NMDA activation, glycine is the major neurotransmitter released by interneurons in the spinal cord. This neurotransmitter acts on ionotropic channels, and these channels behave in the same way as GABA-A receptors do. They are ligand-gated chloride channels. Alright, now that we have covered acetylcholine, glutamate, GABA, and glycine, I want to move on to another category of neurotransmitters, which are called biogenic amines. There are 5 well-established biogenic amines: dopamine, norepinephrine, epinephrine, serotonin, and histamine. To cover biogenic amines, I want to focus on their synthesis pathways and then discuss some particularities they have relative to the other neurotransmitters, as well as what general functions they might have. Let's begin our discussion with the first 3: dopamine, norepinephrine, and epinephrine. Together, these 3 neurotransmitters form what are called the catecholamines. The name catecholamines comes from their structure, which, as you might notice, all 3 have what we call a catechol moiety, which is essentially a benzene ring with 2 alcohols. And then the amine comes from the fact that they all have an amine group that is attached to the ring. As you might suspect from the high similarity in structure, the 3 neurotransmitters are synthesized from a common precursor. This synthesis, which occurs at the presynaptic terminal of the neuron, uses the essential amino acid tyrosine to make the 3 neurotransmitters in 4 steps. To cover the synthesis pathway, I'll first go over each reaction individually, and then we'll come back to see the big picture. To get the synthesis going, the first step of the pathway is catalyzed by the enzyme tyrosine hydroxylase, which converts tyrosine to a compound named DOPA. To do so, tyrosine hydroxylase hydrates, or in other words, adds a hydroxyl group, here in green, on the benzene ring to form the catechol moiety. This reaction requires oxygen, as well as a compound called tetrahydrobiopterin, which will be the source of hydrogens for the hydroxyl group. The addition of OH on tyrosine causes the formation of water as well as the formation of dihydrobiopterin. To complete this picture, the oxidized form is regenerated into the reduced form by the enzyme pteridine reductase and uses NADH to do so. An important point to mention here is that tyrosine hydroxylase is the rate-limiting enzyme in this pathway, which means that the production of the neurotransmitters is highly regulated at the first step. By the way, DOPA stands for dihydroxyphenylalanine, and it is usually the L isoform that is synthesized. In the second step, DOPA gets decarboxylated, or in other words, loses the carboxyl group here in red, and this reaction is catalyzed by the enzyme L-DOPA decarboxylase. The reaction yields dopamine and carbon dioxide. In the third step, dopamine gets hydrated, here in green, by the enzyme dopamine beta hydroxylase to make norepinephrine, also referred to as noradrenaline sometimes. In the fourth and final step, norepinephrine gets converted to epinephrine, also known as adrenaline, by the enzyme phenylethanolamine N-methyltransferase, or PNMT. This enzyme methylates, which means that it adds a carbon group, and it does so on the amine group of norepinephrine. The methyl group to make this reaction comes from S-adenosylmethionine, here shown in light blue on the sulfur, and after the loss of the methyl group, the compound now becomes S-adenosyl L-homocysteine. In a condensed way, the pathway looks a bit like this, where on top you have the substrate names and below the respective enzymes that catalyze each reaction. One important thing to note is that the neurons that do release either dopamine, norepinephrine, or epinephrine do not necessarily have all of the enzymes. For example, a neuron that releases dopamine will only have up to dopa decarboxylase, whereas a neuron that releases epinephrine will have up to PNMT. Alright, now that we have seen the synthesis pathway for catecholamines, let's now see how serotonin and histamine are synthesized. Starting with serotonin, serotonin is synthesized from the essential amino acid tryptophan, and this synthesis is performed in 2 steps, which requires the help of 2 enzymes, tryptophan hydroxylase and 5-hydroxytryptophan decarboxylase. In the first step, tryptophan hydroxylase adds a hydroxyl group onto the benzene ring of tryptophan, and in the second step, 5-hydroxytryptophan decarboxylase removes the carbon dioxide group in gray to yield serotonin. As a side note, serotonin is also often referred to as 5-HT because that is the abbreviation of its molecular name, 5-hydroxytryptamine. In this short pathway, tryptophan hydroxylase is the rate-limiting enzyme. Now, for histamine, this neurotransmitter is made from the essential amino acid histidine through a decarboxylation reaction that is synthesized by histidine decarboxylase. After synthesis, all the biogenic amines we have considered from dopamine to histamine get transported into vesicles by the vesicular monoamine transporter, or simply VMAT, which uses the proton gradient made by the VATPase. After synaptic release, the signal from these neurotransmitters is terminated by a reuptake mechanism that uses a symporter to create a path for neurotransmitters to return in the presynaptic body. This path is coupled to movement of sodium inside the cell. These transporters are specific for the neurotransmitter they transport, so for example, dopamine is moved by the dopamine active transporter, norepinephrine by the norepinephrine transporter, and so on. For your information, a lot of drugs target these transporters, which prevent reuptake mechanisms from properly functioning. 2 good examples of such drugs are cocaine and amphetamines. Now, to complete our picture on these neurotransmitters, let's discuss their general receptors and what general functions they mediate in the brain. I want to mention right away that a lot of the mechanisms of action of the biogenic amine receptors are mediated by G-protein coupled pathways such as the GS, GI, and GQ pathways. So, if you need more information on these pathways, I highly recommend going back to our G-protein discussion as I covered these pathways in great detail. Alright, starting with dopamine, it turns out that when dopamine is released in the synaptic cleft, dopamine only likes to bind to metabotropic receptors. There are 2 main categories of dopamine receptors, the D1-like and the D2-like receptors. D1-like receptors mediate the GS pathway, and the D2 receptors mediate the GI pathway. To quickly summarize the 2 pathways, the GS pathway leads to the activation of adenylyl cyclase, whereas the GI pathway leads to the inhibition of adenylyl cyclase. Adenylyl cyclase is responsible for the production of CAMP molecules from ATP that are then used to activate pKa. The most common substrates of the catalytic subunits for pKa in neurons include voltage and ligand-gated ion channels, proteins on synaptic vesicles, certain enzymes that are involved in transmitter biosynthesis, and proteins that regulate gene transcription. Hence, you can see that the GS pathway increases kinase activity and phosphorylation, but the GI pathway decreases it. Now, in terms of where you can find dopamine in the body, the 2 principal areas that are associated to it are the substantia nigra and the ventral tegmental area, which are 2 regions found in the midbrain of the brainstem. Projections from the substantia nigra are most known for their involvement in the basal ganglia circuit, which mediates certain aspects of movement. Projections from the ventral tegmental area are known for their involvement in the brain's reward circuit, which mediates reward and motivation. Now, when it comes to norepinephrine and epinephrine, the 2 neurotransmitters also bind only to metabotropic receptors. There are 2 main types of these metabotropic receptors, and they are called alpha and beta adrenergic receptors. Norepinephrine and epinephrine can both bind to these receptors, so don't take the colors of these receptors as norepinephrine binds to alpha and epinephrine binds to beta. The two neurotransmitters can bind to both. One thing I'll note right now as well is that norepinephrine and epinephrine are two neurotransmitters that are highly involved in the autonomic nervous system. More precisely, they are fundamental to the sympathetic nervous system, which mediates our fight-or-flight responses. Consequently, norepinephrine and epinephrine participate in functions like heart rate, blood flow, respiration, and so on, which are topics for which I haven't established the groundwork to properly discuss. Hence, I will name the roles for each receptors, but I won't develop too much because it is not the aim of our discussion. Nevertheless, starting with the alpha receptors, there are two subtypes, alpha-1 and alpha-2. Alpha-1 is a GQ-coupled receptor and its activation activates phospholipase C, which cleaves PIP2 into IP3 and DAG to create second messengers that will activate downstream kinases such as PKC and CaMK. The downstream effects of this pathway are excitatory, and it leads to vasoconstriction, pupil dilation, and contraction of smooth muscles to name a few. On the other hand, alpha-2 is a GI-coupled receptor that is usually expressed presynaptically on norepinephrine terminals. The activation of these receptors leads to the activation of potassium channels by the dissociation of the beta and GABA subunit. Potassium channels opening causes potassium ions to leave the cell and thus causes a hyperpolarization. Since neurotransmitter release is dependent on calcium entry and calcium entry is dependent on voltage, the hyperpolarization caused by potassium channels reduces calcium entry and norepinephrine release. It is noteworthy to mention that alpha-2 receptors can be found in the brain and their activation causes sedation. For the beta-adrenergic receptors, there are three subtypes, beta-1 to beta-3, and all of the subtypes are coupled to the GS pathway. Activation of the receptors leads to an increase in CAMP levels caused by adenylyl cyclase, and this will subsequently activate pKa. pKa activation will then lead to an increase in kinase activity and phosphorylation. Beta-1 receptors are mostly expressed in the heart. Their activation leads to an increase in heart rate, contractility, stroke volume, and atrioventricular conduction. Beta-2 receptors are responsible for vasodilation, bronchodilation, and the relaxation of smooth muscles. Beta-3 receptors are found in adipose tissue, and their activation leads to an increase in fat catabolism. To remember their general effects, you can notice that both alpha-1 and beta-1 produce excitatory responses, whereas alpha-2 and beta-2 produce both inhibitory responses. Now, although the two neurotransmitters activate roughly the same receptors, norepinephrine and epinephrine have distinct functions due to their different expression. The main place in the brain where one can find norepinephrine neurons is in the locus coeruleus, which is located in the pons of the midbrain. Beyond what we have discussed on the sympathetic nervous system, the projections coming from the locus coeruleus are involved in sleep, wakefulness, arousal, and attention to name a few. Epinephrine neurons in the central nervous system can mostly be found in the medulla, and their projections go mainly to the thalamus and hypothalamus to regulate functions like respiration and cardiac function. Moving on to serotonin, there are 7 known families of serotonin receptors that can be found in the body, and 6 of them are metabotropic. Out of all the biogenic amines, serotonin is the only one to be known to activate a ionotropic receptor named 5-HT3. This type of ionotropic receptor is very similar to acetylcholine receptors in the sense that it is a pentamer and that upon opening, it is permeable to cations. Hence, this receptor has a reversal potential near 0 mV and causes excitatory postsynaptic potentials, or in other words, depolarizations. When it comes to the metabotropic receptors, the 6 different families mediate the GS, GI, and GQ pathways. The receptors 5-HT1 and 5-HT5 mediate the GI pathway, whereas the receptors 5-HT4, 5-HT6, and 5-HT7 mediate the GS pathway. Finally, the 5-HT2 receptors mediate the GQ pathway. These pathways are the exact same as we have seen previously. The main region in the brain where serotonin neurons can be found is located in a structure named the Rafe nuclei, and serotonin is implicated in a wide diversity of functions like wakefulness, motor behaviors, emotions, and hunger. It is noteworthy to mention that serotonin also has some projections to the spinal cord to modulate sensory, motor, and autonomic functions. Finally, when it comes to histamine, all of its known receptors are metabotropic. There are 4 main families, and just like in the case of serotonin, these metabotropic receptors carry out the GI, GS, and GQ pathways. Receptor H1 mediates GQ, H2 mediates GS, and H3, H4 mediate GI. In terms of its location in the brain, histamine is mostly found in neurons of the hypothalamus, and their connections mediate wakefulness and attention. In summary, of all we've discussed about biogenic amines regarding the receptors and functions, you can refer to this to get a general overview. Alright, with biogenic amines now covered, let's move on to the next type of neurotransmitter I want to cover. This class of neurotransmitters is referred to as the purines, and it includes adenosine triphosphate, more commonly referred to as ATP, and adenosine, which is the degradation product of ATP, or in other words ATP, without its 3 phosphates. It turns out that all synaptic vesicles have some levels of ATP. Accordingly, ATP is always getting released during synaptic events. At synapses where it operates, ATP can bind to ionotropic and metabotropic receptors. The ionotropic receptors for ATP are named P2X and are essentially non-selective cation channels. This means that their reversal potentials are at around 0 mV and cause excitatory postsynaptic potentials. An example of where these channels might be found is in sensory nerves, where they play roles in touch and pain pathways. The metabotropic receptors for ATP are named P2Y. There are several subtypes of P2Y from 1, 2, 4, 6, and 11, 14 that mostly mediate GQ and GI pathways respectively. Now, when it comes to adenosine, they activate metabotropic receptors in the P1 family, which is subdivided in 8 types. A1 and A3 activate GI GPCRs, and A2A and A2B activate GS GPCRs. Adenosine and ATP together mediate an immense array of functions, so even a concise list wouldn't do justice to them, as they are widely distributed in the brain, heart, kidneys, and adipose tissues. Alright, with the classic neurotransmitters like acetylcholine, glutamate, GABA, glycine, the biogenic amines, and the purinergic neurotransmitters now covered, I want to go over some neurotransmitters that are a bit unconventional relative to what we're used to seeing. These neurotransmitters are endocannabinoids and nitric oxide, and they are often referred to as transcellular messengers for reasons you will soon understand. To see how these neurotransmitters work, let's begin with endocannabinoids. First of all, the reason why endocannabinoids are called transcellular messengers is because they mediate what is known as retrograde transmission. When we consider the synapse between the presynaptic and the postsynaptic partner, endocannabinoids are usually synthesized at the postsynaptic terminal, and because they are hydrophobic, they are able to cross the membrane to bind to receptors that are located on presynaptic sites. The activation of these receptors then activates all sorts of stuff that impacts both the presynaptic and postsynaptic cells. There are 2 well-known endocannabinoids that are synthesized in the body, anandamide, which is abbreviated as AEA, and 2-AG. In the case of 2-AG, the synthesis of 2-AG is initiated by the activation of postsynaptic G proteins that activates phospholipase C. Remember that one type of G protein that activates phospholipase C is the GQG protein. Phospholipase C then goes on to hydrolyze a membrane-bound lipid by the name of phosphatidylinositol to produce diacylglycerol or DAG. One thing that I want to point out really quickly is that IP is very similar to PIP2, the molecule that was hydrolyzed in the GQ pathway, and as you can see, the main difference is in the inositol group, which has 2 additional phosphates in IP3. Now, the diacylglycerol made in this instance then gets converted to 2-AG by diacylglycerol lipase. The newly made 2-AG can now exit the postsynaptic terminal due to its hydrophobic properties and go bind to its associated receptor. Before we move on to talk about the receptors, let's see how anandamide is synthesized. The synthesis of anandamide is initiated by a rise in intracellular calcium concentration that activates phospholipase D. Active phospholipase D then cleaves a molecule that is abbreviated to NAP to form anandamide. Here again, when anandamide is made, it is able to leave the cell and go bind to its receptor. The receptors for endocannabinoids are all G-protein coupled metabotropic receptors, and there are two major known types, CB1 and CB2. CB1 receptors are most often found in the brain on presynaptic terminals of GABA or glutamatergic synapses. CB2 receptors, on the other hand, are found mostly on lymphocytes where they mediate immune responses. So given that we're discussing neurons, I'm not going to dive any deeper in them. I'll focus on CB1 because it is the one that is mostly expressed in the brain and most relevant for our inquiries. The CB1 receptor is mostly known for activating the GIG protein, which causes the inhibition of adenylyl cyclase and thus the inhibition of CAMP production. The reduction in CAMP molecules leads to a reduction in net phosphorylation due to reduced pKa activity. Moreover, CB1 activation reportedly activates MAP kinase, inhibits voltage-gated calcium channels, and activates potassium channels. The reduced activity of calcium channels, paired with the increase in outward current from potassium channels, causes the reduction of neurotransmitter release. Indeed, since neurotransmitter release is dependent on calcium and calcium is dependent on voltage, all of these effects mediated by the CB1 receptor cause the presynaptic terminal to release less. At synapses that release GABA, this phenomenon is known as depolarization-induced suppression of inhibition, and inversely, in glutamate synapses, it is known as depolarization-induced suppression of excitation. This phenomenon is very important when it comes to synaptic plasticity, which is a topic we'll cover shortly. After receptor activity, the endocannabinoids are carried back into the postsynaptic cell by carriers and get hydrolyzed by an enzyme by the name of fatty acid amide hydrolase. The products of this hydrolysis then get recycled or go into other lipid pathways. To finish our discussion on endocannabinoids, you might have noticed that the word itself, endocannabinoid, is very similar to cannabis, which is a word that we hear often. It turns out that cannabis is an exogenous cannabinoid, meaning that it comes from outside the body, whereas endocannabinoids are synthesized inside the body. Two examples of famous exogenous cannabinoids are THC and CBD, which can bind to the CB1 and CB2 receptors. Given that these compounds come from plants, they are also called phytocannabinoids. Moving on to the next transcellular signal, we have nitric oxide. In comparison to the other neurotransmitters we have covered, nitric oxide is very interesting because it moves in the cell under a gaseous form, and because of that, it can easily cross membranes to target nearby cells. The synthesis of nitric oxide is made as a side product from the conversion of arginine to citrulline by nitric oxide synthase. After synthesis, nitric oxide can interact and activate a cytoplasmic soluble guanylyl cyclase. Guanylyl cyclase is very similar to adenylyl cyclase, where its activation produces cGMP molecules from GTP. The newly made cGMP molecules then go on to activate the cGMP-dependent protein kinase, also known as PKG, and just like pKa, PKG goes on to phosphorylate some substrates that then affect the cell in various ways. The structure of PKG is very similar to pKa, in the sense that it has catalytic and regulatory domains, but the one difference is that PKG is a single polypeptide and its catalytic domains do not dissociate upon cGMP binding. The lifespan of nitric oxide is not very long, however. It is a very reactive gas and ends up reacting with oxygen to produce various nitrogen oxides that are inactive. In summary, transcellular transmitters are said to be unconventional because they are not released in vesicles by exocytosis, but instead they cross the membrane directly to affect other cells, thereby acting as retrograde transmitters. Now, based on the list we have here, we have covered some of the most important neurotransmitters, except for one very large family called the neuropeptides. Neuropeptides are small-chain amino acids that bind to GPCRs to mediate an immense array of functions, not to mention the fact that there are over 100 known neuropeptides. Because of their incredible diversity in type and in function, I will not cover them in this video and I will likely cover them in their respective contexts. Nonetheless, if I were to cover them like I did for the other neurotransmitters, a lot of their mechanisms from the release to receptor activity would be very similar to topics we have covered. In all, The main neurotransmitters in the brain, except for neuropeptides, look a bit like this. Alright, now back to the schematic where we've established the differences between the neuromuscular junction and the neurons in the central nervous system. We have two final but very complex differences to discuss, which is the fact that the postsynaptic cell receives many inputs, and these inputs are generally weak, such that it takes many excitatory inputs to generate an action potential in the postsynaptic cell. These two statements seem rather simple and encompassing, but it hides a few key aspects that are fundamental to neurons. If we recall the neuromuscular junction, remember that when the motor neuron fired and released acetylcholine on the muscle fiber, it always caused an action potential that then caused the contraction of the muscle. This event is essentially a one-to-one. The motor neuron fires and the muscle contracts, which is rather intuitive and follows how we expect muscles to behave. Hence, the communication between the motor neuron and the muscle fiber is as straightforward as it can be. In the central nervous system, neuronal communication can become highly complex because the weak inputs might not always be sufficient to reach action potential threshold. In any case, neurons must add the inputs they receive to reach that threshold. This aspect is often referred to as summation. As you might remember from our discussions on the action potential is most often initiated at the axon initial segment. The reasoning behind why this place is so important in deciding whether the neuron fires or not is because it has the lowest action potential threshold across the neuron. Remember that the definition of the threshold, as defined in our action potential discussion, is the membrane potential at which the inward current, which is mostly carried by sodium, outweighs the outward current from potassium and/or chloride. It turns out that the axon hillock is the place which usually has the highest density of voltage-gated sodium channels and thus provides the most inward current to the cell. Another important point to remember is that before the neuron hits the threshold, the signals in the cell are all flowing passively, which means that they decay over time and are not self-sustained like the action potential. For that reason, when the cell adds the inputs together to reach threshold, this summation can be done both in time and space, which introduces us to the concept of temporal and spatial summation. Let's start with temporal summation, which is defined as the summation of two or more consecutive signals. To explain this principle, I want to reconsider the passive membrane properties that I have introduced previously, such that I can better explain some new concepts. If I go too quickly on these concepts in this discussion, I recommend watching the videos on the equivalent circuit model and the RC neuron to get additional insights on these concepts. Alright, we've established that there are three fundamental passive membrane properties which govern the temporal and spatial time course of passive propagation. The three properties are the membrane capacitance, the membrane resistance, and the axial resistance. To give a brief description for each, let's start with the membrane capacitance. This property arises by the separation of charges between the membrane. The separation of charge, which can be labeled as delta Q, creates a voltage difference delta V due to the electric field between the charges. This voltage difference is what we know as the membrane potential, and it is related to delta Q by the membrane capacitance constant. Another consequence of charge separation is the fact that when charges enter the neuron, here in this instance positive charges, it causes charges on the opposite side to feel a repulsion and move away. This charge movement creates a current which we've called the capacitive current IC. From delta Q equals Cm delta V, we can derive the equation of the capacitive current if we consider the definition of the current, which is simply the derivative of charge over time. Now, the second property is the membrane resistance, which arises from the fact that the membrane has ion channels that let ions flow in and out of the cell. For our temporal summation purposes, there are two distinct types of channels that we need to take into account: leak channels and the ligand-gated synaptic receptors. As we've discussed in the equivalent circuit model video, ion channels obey Ohm's law, and thus the current that flows through them is equal to the voltage difference divided by the membrane resistance. Another important note about them is that the movement of charges through these channels is powered by the electrochemical gradients of the ions that flow through them. Accordingly, we've derived a modified version of Ohm's law that encompasses all of these different properties. Note that G here stands for the conductance, and it represents the inverse of the resistance. Now, in terms of their roles, leak channels are the channels that are open at rest and govern the resting membrane potential. Their summed resistance or conductance, depending on how you see it, is known as the leak resistance RL. This value is also sometimes referred to as the input resistance. When it comes to synaptic channels, they are the ones that only open during synaptic events. It is very important to keep in mind that their opening changes the total resistance of the cell, but this is a detail I'll come back to shortly. The third and final property is the axial resistance, and it corresponds to the internal resistance that charges feel when they move inside the dendrites or axons. The equation that models this property is essentially the same as the equation that models electron flow in electrical wires. Now, to understand temporal summation, we only need the two first properties, membrane capacitance and membrane resistance. Another tool that will be relevant for us to understand how temporal summation occurs is to have the equivalent circuit model of the synapse. This circuit basically models the neuron as an RC circuit, which is very convenient for us because from that we will be able to determine equations that will allow us to see how the membrane potential changes as a function of time based on our passive membrane properties. Now, in our system, there are two dynamic states to consider: during the synapse when the synaptic channels open, and after the synapse when the neuron returns to baseline. Let's start with during the synapse. To find the equation of the membrane potential as a function of time, we can establish that the current as a function of time during the synapse will be the sum of the current coming from the capacitor and the resistors. Next, we can substitute the currents for the capacitor and the resistor by their definitions. Here it gets a bit tricky because, as I mentioned a few moments ago, synaptic channels will be active during the synapse, and for that reason they will affect the resistance of the system. Based on electric circuit laws, we can find the total sum of the resistances by taking the inverse of the sums of the inverse of each resistance. I will note this new resistance that occurs during the synapse as RSL. With this new resistance, we can now continue the derivation of the equation by multiplying each side by the presynaptic leak resistance. From here, we can define two new quantities, which are the steady-state potential and the time constant. You will later see their relevance when we go over them in a bit more detail, but for the moment, they allow us to make the equation less clustered. For those of you who have watched the RC neuron video, I changed the notation of Vmax to VSS because I feel like VSS, which stands for the steady state, is more telling on what VSS is rather than Vmax. Nevertheless, by substituting these variables, we can rearrange the equation to get this form. The next step in our derivation will actually be to integrate the differential equation we came up with. I will let you go through the steps on your own, but basically we end up getting this exponential equation where the membrane potential as a function of time is equal to the steady-state voltage times the exponential factor. This should suffice us for the first state. Now, when the synapse is closed and the membrane potential returns to baseline, the conditions change. First, the current as a function of time now equals 0 because the currents are only mediated by the capacitor and the resting resistors, which pass current to return to the resting state. But there is no additional source of current now. Another important distinction is that since the synaptic channels are now closed, we only have to take into consideration the resistance of the leak channels, RL. From there, the derivation follows the same steps as before and yields us this equation. You will notice that since the added current in the system is now equal to 0, there is no steady-state potential to consider. Instead, since this decay picks up right after the end of the channel's opening, we now have to consider the membrane potential at time 0, but I'll come back to that aspect very shortly. At the end, you will notice that the two equations are very similar to each other, but there are two key differences. First, the equation that models the membrane potential when the synaptic channels are open describes an exponential growth, whereas the equation that models the state when the synaptic channels are closed describes an exponential decay. Secondly, when the synaptic channels open, we need to take into account the resistance of the synaptic channels, and that is reflected in the value of the steady-state voltage and the time constant. Whereas, when channels close, it is only the leak resistance that is to be taken into account. Because of this, the growth and decay will have different time constants and thus different time courses. By the way, due to the fact that we add the inverse of the resistance, the total resistance will always decrease when we add a new resistance. Hence, the time constant when the synaptic channels are open will always be smaller than when the channels are closed. With this being said, let's now get into temporal summation and see how what we just derived helps us understand that aspect. To exemplify temporal summation, I want to give a numerical example of what a problem might look like because I believe that's more telling and better for our comprehensions. So, let's imagine that the membrane of this postsynaptic cell receives an excitatory connection that fires twice at 100 Hz. We want to figure out what will be the membrane potential at the end of the second pulse, and we will assume that the currents are constant. Here, we make this assumption to keep the steady-state potential constant, as you will see later. We'll also imagine that based on the values of the resistance and capacitance of our postsynaptic neuron, the time constant during the synaptic activation is 1 millisecond, and when the channel's closed, the time constant becomes 3 milliseconds. Furthermore, we'll imagine that the current injection from the channels lasts 7 milliseconds and that the steady-state potential is about 10 millivolts. To better visualize what's going on, let's also plot the membrane potential and the synaptic current as a function of time. Okay, so to solve this problem, I suggest to first understand when the synapse is active or not based on the frequency and the injection time. As a side note, the use of frequency to describe temporal dynamics in neurons happens very often. As a reminder, frequency, which is measured in hertz, corresponds to the inverse of the time interval between each pulse. In our problem, a frequency of 100 Hz means that the interval between each pulse is therefore 10 milliseconds. With this information, we can see that the first pulse lasts 7 milliseconds, and 3 milliseconds after its end, another one is fired for 7 milliseconds again. In the plot of the membrane potential, we can also plot the steady-state potential, which follows the same shape as the current. Now that the time frame is established, we can use our exponential equations to find the final membrane potential. From 0 to 7 milliseconds, plugging our numbers into the growth equation gives us that the membrane potential at 7 milliseconds will be plus 10 millivolts. In cases like this, where the time elapsed is significantly larger than the time constant, about 5 times tau, we can usually say that the membrane potential has reached the steady-state potential. For that reason, the time constant is a good indicator to approximate where the membrane potential actually is in relation to the steady state. If, for example, the time constant was 30 milliseconds, well, we would have known that just by eyeballing the equation that the depolarization is far from the steady state. Moving on, we now need to compute by how much the signal decays in the 3 millisecond interval between the end of the first pulse and the start of the second. By plugging the numbers, we get that the signal decays to 3.7 millivolts. In cases like this, where t equals tau, there is another trick that can help you save some time. Indeed, when time and tau are equal, the exponential factor simply becomes e to the negative 1 or 1 minus e to the negative 1, which respectively gives 0.37 and 0.63. Hence, here I only had to multiply 10 by 0.37 instead of entering the exponential term in my calculator, which can be a bit tedious at times. Finally, we can add the membrane potential at the end of the second pulse by adding 10 millivolts to 3.7 to give a final answer of 13.7 millivolts. In a case like this, where the time constant and steady-state potential stays the same for each pulse, there is no need to compute the depolarization again. We can just take what we found in the first step. I hope this problem wasn't too difficult to follow through. If you have any questions, make sure to leave them in the comments. To end our discussion on temporal summation, I want to briefly discuss the time constant. As we've seen in the problem, the time constant gives us a good estimate of where the decay and growth parts might end up. Now, if in this problem, the open and closed time constants were something small, let's say 0.1 millisecond and 0.3 milliseconds, you can see that the results are drastically different. Indeed, the time constants for the growth and the decay are so quick, relative to the current injection, that no summation can even occur. This means that to reach the threshold in these conditions, the presynaptic cell would need to fire at considerably higher frequencies or inject very high depolarizing pulses. On the other hand, if in the problem the open and closed time constant were very large, let's say 10 and 30 milliseconds, there would be some summation, but the depolarizations only reach a fraction of the steady-state potential. For that reason, there is a certain trade-off between low and large time constants. If they are too small, then the potentials will rise quickly to the steady state, but it also means that the potentials will decay quickly and prevent summation. On the other hand, if the time constant is too large, it takes too much time for the postsynaptic potential to reach the steady state that it only reaches a certain fraction of it, but at least the slow decay increases the likelihood for consecutive signals to summate. Hence, we can say that as a rule of thumb, neurons that have longer time constants are more likely to summate in time because longer time constants cause longer decay times and thus more time for signals to summate. Alright, now let's move on to spatial summation, which occurs when two or more synaptic potentials are added in space. In temporal summation, we focused a lot on the time constant, and as you can imagine, here we will focus a lot on the space constant. Now, although they are two different measures, they can be interpreted in pretty much the same way. By the way, if ever you are unfamiliar with the space constant, I highly suggest you to watch this video on the cable theory model of the neuron, where I thoroughly explain how the constant can be derived and what it means. From the video, we covered that signals decay as a function of space, and that a high value of the space constant means it decays slower relative to a smaller value. Hence, if we imagine two different pulses that are activated at the same time, the likelihood that the two will be summed together will be much greater if the space constant is higher, since the signals will have decayed less. In summary, there are two main types of summation that we can consider: temporal and spatial. The main elements that can give us the most insight as to the signal summation are the time and space constants. In general, the bigger they are, the less the signals decay and the more probable it is they will summate. One thing to keep in mind here is that the two forms of summation always happen at the same time. It is only in our idealized theoretical worlds that the two can be analyzed separately, but in real neurons, the synaptic connections get integrated in time and space simultaneously. Also, I looked at summation through the lens of excitatory connections, but keep in mind that inhibitory connections also get summed and participate in the signals the neuron receives. Alright, back to our schematic on the differences between the neuromuscular junction and the central nervous system. We now have covered metabotropic receptors, the different neurotransmitters, and synaptic summation, which already gives us a pretty good idea on how neurons in the central nervous system are different from neurons in the neuromuscular junction. The final aspect that I want to cover now is a bit hidden under the lines of the differences shown here. As I mentioned multiple times, neurons in the central nervous system experience very weak inputs, but in very large quantities, whereas the neuromuscular junction is essentially one large input on the muscle. This difference, as we've seen, leads to the concept of synaptic integration, but it also leads to the idea of plasticity. Basically, due to the fact that the inputs are so small in the central nervous system, it leaves room for them to be modulated, so to increase or decrease their strength, which is known as plasticity. The notion of plasticity is fundamental to the well-being of our nervous system because it is through these mechanisms that we learn and remember. Now, when we think about a presynaptic neuron communicating with a postsynaptic partner, the signal propagation between the two is dependent in both the presynaptic and postsynaptic partners. Indeed, to appropriately relay the message, the presynaptic cell must exocytose its neurotransmitters such that the postsynaptic receptors can transduce it into a passive signal that might reach the threshold. If, for some reason, the presynaptic input is decreased, maybe there is an issue with release of transmitters, or there are no more vesicles, or whatever it might be, the diminished quantity of neurotransmitter release will impact the signal made on the postsynaptic cell. Also, the converse is true. If there are more transmitters released, then the response will be bigger. We can also imagine on the postsynaptic cell, if there are more or less receptors to pick up the signal, the response will also be modulated accordingly. Hence, plasticity can occur on either the presynaptic and/or the postsynaptic side. Before we jump into the different mechanisms of plasticity, I want to mention right away that a sizable portion of plasticity mechanisms are rooted in the probabilistic nature of the synapse. For that reason, I want to begin our discussion by first describing the binomial model of the synaptic release, which we will later use to help us better interpret certain aspects of plasticity. If you recall from our discussion on the neuromuscular junction, you might remember in the experiment carried by Katz, as well as Boyd and Martin, that the release of unitary vesicles was entirely stochastic, or in other words, probabilistic. In the framework of this experiment, The bottom line was that by using a probabilistic model, we were able to support the idea that each vesicle were a unit because they had a fixed amount of depolarization that they produced on the postsynaptic cell. Now, knowing that, or at least having strong supporting evidence that synapses act in a probabilistic manner, I want to use a simple probabilistic model to create a tool for us. The probabilistic model in question that I will use is the binomial distribution. If you are not familiar with this distribution, let me give you a brief rundown of its main elements. To do so, let's first consider a simple conceptual example. Imagine that in a bowl, there are 3 red balls and 4 blue balls. What will be the probability P to pick up 2 red balls in 3 independent trials? To solve this problem, let's first establish some variables. We have 3 independent trials, which we will call N, 2 successes within these trials, which we will call X, and from the context that we have here, we know that out of the 7 total balls, there are 3 red balls, so the probability to pick up a red ball in a given event is 3 out of 7. This probability is known as the probability of success, and it is important that it remains constant. It turns out that we can model our variables and determine the probability of X successes in N independent trials with this equation, which represents the binomial distribution. Also, the expected value of this distribution is equal to the number of trials N times the probability P. When we apply it to our problem, we get that the probability to pick 2 red balls out of 3 trials is about 30%, and that we expect a value of about 1.29. The expected value in our case means that we should expect to pick 1.29 red balls in 3 trials. Now, it doesn't really make too much sense here as decimal balls don't exist, but it will be important shortly. Finally, I want to touch quickly on this term here, which reads as N choose X. This is an important factor in our equation because it corresponds to the number of ways there are to choose a red ball X times out of N. In our example, there are 3 ways. Now that we understand this, let's apply that knowledge to a synapse. Let's start from the equation we had in our conceptual problem and redefine the variables. The probability P of success in our case here will be the probability of vesicles being released, which we will call P vesicle. We will assume that the event of vesicle release is all or nothing, and its probability remains constant. Now, when it comes to N, instead of thinking in terms of trials, we can think in terms of quantas that are released. It turns out that in the central nervous system, it is not uncommon to impose the limit of quantas to the number of release sites. In our left diagram, N is currently equal to 1, but if an additional release site was to be added, then N can equal to 2, or if another one is added, then N can equal to 3. This sort of assumes that each release site releases 1 quanta only. Otherwise, we could have modeled N as being the number of quanta released per site, so 1 in this case or 3 in this one. To avoid any complications, I will stick to the release sites as it makes it a bit more intuitive in my opinion. For X, it will be similar to the conceptual example. It will correspond to the number of vesicles released within the trials. The final variable that we will consider will be the quantal amplitude of a vesicle, and it will be used to multiply our expected value to scale it in units that we are used to seeing like voltage or current. This is especially important if we want to test our model in experimental conditions, because the recordings in the laboratory will be made in voltage or current units. Now, let's do a quick example to see how these variables operate. Imagine a synapse with only one synaptic site, so n equals 1. If the probability of release is 0.4 and the quantal amplitude is 2 millivolts, what will be the probability to release one vesicle? Also, what will be the average response at this synapse? In this problem, since we only have one site, well, the probability to release one vesicle will be 0.4 accordingly. For the average response, we can use our equation of the expected value and determine that the average response would be about 0.8 millivolts. This example was rather straightforward, so let's see another one that might be a bit more challenging. What will be the probability to release 0, 1, 2, and 3 vesicles if there are 3 synaptic sites? Again, what will be their average response at this synapse? To do this problem, I want to introduce a new variable named P failure, which corresponds to 1 minus P vesicle. To make it more convenient, we will call this variable Q. Now, by using the binomial equation, we can find the probability for each scenario. For example, the probability that no vesicles will be released is 0.216, the probability that one will be released is 0.432, and so on up to 3 vesicles. In instances like this, where we have many synaptic contacts, the combination term is very important because it presents the different possible combinations of the scenarios that can occur. For example, for one vesicle released, it can either be the left one, the middle one, or the right one that fires. Hence, there are 3 possibilities for firing, and the same logic applies to the other cases. When it comes to the average response, we can use our equation to arrive at the answer of 2.4 millivolts. There is also an alternative way to get this response, and it is by summing the product between the probability and the quantal amplitude at each site. This will give us 2.4 millivolts as well. Now, the case with multiple synaptic sites can be a bit tricky to understand what is going on, and to help ourselves, we can refer to a graph to have a better intuitive idea. On this graph, we will plot the probability for each scenario as a function of the quantal amplitude. So for x equals 2, for example, we have a probability of 2.88, and it is associated with a quantal amplitude of 4. You will notice that from this graph, we can get the probability of a certain event from the area under the curve, and we can also get the number of synaptic contacts just by looking at the non-failure peaks. Since there are 3 non-failure peaks, we know that our synapse has 3 presynaptic terminals. Finally, in cases with multiple synaptic sites, so when n is over 1, we can establish a new variable called p-synapse, and this variable corresponds to the summed probability of released vesicles. In our case, this probability equals to 0.784. We can relate p-synapse with p-vesicle in this relation. To make sure you understand what is going on, I suggest you to try and derive this relation. All the necessary information to do so is shown here. Alright, now that we understand this binomial model, let's begin our discussion on plasticity. A fundamental aspect of plasticity is that it can happen on many timescales. The 2 broad ways to classify these timescales is through the short-term and the long-term. Accordingly, let's begin with short-term plasticity, which corresponds to events that last on the scale of a few minutes or less. Generally speaking, short-term plasticity occurs on the presynaptic terminal, and the 2 main forms are called paired pulse facilitation and paired pulse depression. To see how plasticity occurs, we always need to consider a baseline response, so pre-plasticity and post-plasticity response. For that reason, we will need at least 2 consecutive pulses delivered to the presynaptic terminal to be able to see any changes in the synaptic strength. Alright, let's begin with paired pulse facilitation. To see how it occurs, let's plot the membrane potential of the postsynaptic neuron and the current that will be injected in the presynaptic cell. When 2 quick successive pulses are sent, paired pulse facilitation results in the second depolarization on the postsynaptic cell to be larger than the first one. Based on different studies, the leading hypothesis on how this might occur is from residual calcium accumulating in the presynaptic cell, which causes more vesicles to be released. This phenomenon is also believed to occur mostly at low release probability synapses. Let's see how. First, remember that on presynaptic terminals, there are generally 2 communities of vesicles, the ones who are ready to be released, which form the readily releasable pool, and the ones that are in the reserve pool waiting to be docked at the membrane. When the first pulse comes and depolarizes the terminal, a certain amount of calcium, here shown in blue, enters the cell, and a fraction of it is used to release vesicles, which generates the first response in the postsynaptic neuron. The other fraction of the calcium remains in the terminal, and when the second pulse arrives shortly after, it adds on calcium that causes even more neurotransmitters to be released. Hence, the second pulse becomes bigger because more receptors get activated. In this scenario, the fact that the synapse has a low release probability is important because it ensures that the presynaptic terminal has the necessary amount of vesicles ready at the edge of the membrane to be released on the second pulse. Now, for paired pulse depression, the second pulse causes the response on the postsynaptic cell to be smaller than the first one. This form of synaptic plasticity is believed to occur from vesicle depletion in high-release probability synapses. At these high-probability synapses, the first pulse causes the readily releasable pool to exocytose very quickly most of the vesicles it has, so when the second pulse comes, there are less vesicles that are ready to be released, and that causes the second postsynaptic response to be lower. You will notice that there is a very important time dependence to these 2 mechanisms. If the second pulse is delayed too long relative to the first, then the calcium would have time to dissipate and the vesicles would have time to return. So, the timing of the second pulse is critical for these types of plasticity to happen. Also, as I said, since the vesicles can reform quickly and the calcium gets buffered quickly as well, it explains why these two forms of plasticity act on the short term. In mathematical terms, we can quantify the amount of paired pulse facilitation or paired pulse depression from the paired pulse ratio and paired pulse facilitation metrics. In the paired pulse ratio, we take the ratio of the second amplitude over the first. In cases of facilitation, the ratio will be above 1, and in cases of depression, the ratio will be below 1. For the paired pulse facilitation metric, we do the same thing, but in the numerator we subtract the amplitude of the first response. If the value is positive, it corresponds to facilitation, and inversely, if the value is negative, it corresponds to depression. Now that we understand a bit more how plasticity occurs in the short term, let's explore the long-term aspect. To do so, we will consider an important area in the brain named the hippocampus that has been instrumental in the discovery and the development of synaptic plasticity. To find the hippocampus, we can take a coronal section at the level of the temporal lobe. The hippocampus will be located in this external portion of the brain on either sides. Now, the hippocampus is a very important region that mediates a lot of functions like memory, and there is a lot of work done there to support this idea, but for our purposes, we will simply use the hippocampus to introduce some anatomical landmarks that will help us discuss plasticity. Concepts that relate to the connections of the hippocampus and how it mediates memory are topics that will be covered in other videos. Now, when it comes to the circuitry of the hippocampus, we must first note that it has three important regions with distinct cells, the CA1 and CA3 regions, which both have pyramidal neurons, and the dentate region, which has granule neurons. The input to the hippocampus comes from what is known as the perforant pathway, which makes connections on granule cells as well as CA1 cells. Granule cells in the dentate region synapse to CA3 cells through the mossy fiber pathway, and the CA3 cells synapse on CA1 neurons through the Schaffer Collateral pathway. This completes the basic circuitry of the hippocampus. I want to note that textbooks will often flip this diagram for some reason, and most likely you have seen the hippocampus in this orientation, but since we do not really consider the anatomical implications of the hippocampus, I will use the unflipped orientation since it is more faithful to the coronal section. In this circuit, two general forms of long-term plasticity have been discovered: long-term potentiation, or LTP, and long-term depression, or LTD. Let's begin our discussion on long-term plasticity by first discussing LTP and what experiments allows us to see it. For that, we will consider the Schaffer Collateral synapse of the CA3 neurons onto the CA1 neurons. Just so we set the anatomical basis, the synapse between the CA3 and the CA1 neurons is mediated by glutamate and is thus excitatory. It is important to note as well that this connection is formed on the dendritic spines of the CA1 neurons. Now, the basic setup for plasticity experiments in such scenarios is to electrically stimulate the presynaptic cell and to record the response from the postsynaptic cell. The EPSP measurements measured at the postsynaptic cell are then plotted across time. When electric stimulations are done at a low frequency, let's say once or twice per minute, the amount of depolarization recorded on the postsynaptic cell is constant and passive. You will notice that the EPSP axis is normalized to the baseline, and the baseline corresponds to this first period when the cell is stimulated at low frequencies. Hence, the first responses that we see here are at 100%. Now, when one induces a very high frequency train of electrical stimulation in the presynaptic cell, also known as a tetanus, it causes the postsynaptic cell to fire. After the tetanus period, the size of the postsynaptic response when the neuron is stimulated again is bigger than what it was during the baseline period. As you can see, the increase in response gets stabilized over time, but the postsynaptic response remains bigger than baseline a long time after the tetanus. In such a plot, we can compute the percent amount of plasticity that has been caused by the tetanus by this equation. Now that we understand how to interpret the basic plasticity recording plots, let's see how we can understand the mechanisms that underlie LTP at the Schaffer Collateral. It turns out that LTP at the Schaffer Collateral is highly dependent on NMDA receptors. If NMDA receptors seem foreign to you, Recall that they are ionotropic channels of glutamate that we've covered in our discussion on different neurotransmitters. One important aspect that we have established in this discussion that can help us prove the relevance of NMDA receptors is that the compound AP5 acts as an antagonist to the NMDA receptors. Hence, when we perform tetanization experiments with AP5 blocking the NMDA receptors, there is no increase in postsynaptic response, and thus entails that NMDA receptors play an important role in mediating LTP. Two key properties of NMDA receptors that make them such a unique receptor and explain how they can mediate plasticity is that first, their opening requires glutamate binding and also postsynaptic depolarization to remove a magnesium blocker, and second, they are able to conduct calcium inside the cell when they open. As we've seen time and time again in our discussion, calcium is a very important second messenger in neurons. If you recall the GQ pathway, you will remember that some of calcium's downstream effects are the binding to calmodulin and the activation of distinct kinases such as PKC and CaMK2. The activation of these two kinases leads to two important aspects of plasticity. First, some of their downstream phosphorylation targets are important proteins involved in the transport of AMPA receptors in and out of the membrane. Accordingly, the phosphorylation of these proteins causes AMPA receptors to be moved from internal structures where they are stored to the membrane of the postsynaptic terminal. With now more ionotropic AMPA receptors, the postsynaptic response is considerably increased as more inward current can enter the cell. The second important target for plasticity are the receptors themselves that can get phosphorylated. This phosphorylation causes the channels to open more and let more current enter. In this entire mechanism, there is an important distinction to make between the induction and the expression of plasticity. Induction corresponds to the biochemical cascade that is activated by the tetanus, and the expression corresponds to the actual long-term changes, so in this instance, the addition of AMPA receptors. Now, this form of LTP that I have described lasts only a few hours, and it has thus been called early LTP. However, there are some forms of LTP called late LTP that can last even longer, but they require gene expression and protein synthesis to happen. To get protein synthesis started in plasticity experiments, remember from our discussions on kinases that kinases like pKa or MAPK can phosphorylate transcription factors that can then go into the cell nucleus and start the synthesis of proteins. One common example of such a transcription factor that is important in late LTP is CREB, which comes from the phosphorylation of pKa and MAPK. To get this process started, it turns out that the complex that calcium and calmodulin form can go on and activate a specific type of adenylyl cyclase. Then, this adenylyl cyclase causes the conversion of ATP to CAMP, which activates pKa. pKa can then go on and activate MAPK and CREB. With sufficient protein synthesis in late LTP, it can eventually lead to the formation of new synaptic contacts, which again will increase the strength of the connection but now for larger timescales, so days, months, or years. Now that we have a good idea of how LTP occurs at the Schaffer Collateral, I want to establish some properties that LTP at the synapse has because it is relevant in the grand scheme of the field of plasticity and memory. First off, I want to mention that when we measured the baseline responses before the tetanus was applied, these responses were sub-threshold EPSPs, and they were not sufficiently strong to generate a change in synaptic strength. To get LTP to occur, it is required that the presynaptic and postsynaptic partners both get very depolarized within a short amount of time. In an experimental setting, we can achieve this by stimulating the CA3 and CA1 neurons together. When we consider a control pathway that is not stimulated along with the stimulated pathway, we can notice that LTP only occurs at the site where both the presynaptic and postsynaptic sites were stimulated. This property is known as input specificity. To explain input specificity, we can think back to the NMDA receptor mechanism. Indeed, since the NMDA receptor requires both the binding of glutamate and a postsynaptic depolarization, only the pathway where both of these requirements are met will cause LTP to occur. Now, in more physiological settings, the postsynaptic partner obviously does not get stimulated by an external electrode. Hence, to achieve that postsynaptic depolarization that the NMDA receptors need, many pathways need to cooperate together to reach that threshold. This property of distinct pathways to help each other out is referred to as cooperation. Tied to this idea of cooperation, there was a very influential postulate written by Donald Hebb that said, when an axon of cell A is near enough to excite a cell B and repeatedly or persistently takes part in firing it, some growth process or metabolic change takes place in one or both cells such that A's efficiency, as one of the cells firing B, is increased. Basically, what he is saying in this quote is that when a group of presynaptic cells that on their own cannot make the postsynaptic cell fire manage to make it fire, then these connections should be strengthened. Accordingly, this postulate is often coined as cells that fire together, wire together. From this postulate, you will notice that the temporal ordering from pre to post is very important, otherwise the connection will not be strengthened. This leads to the idea of a coincidence between the pre and the postsynaptic cell, which, as we've seen, is mediated by the NMDA receptor. Along with cooperativity, another fundamental property of LTP that can occur from the result of multiple pathways interacting with each other is the concept of associativity. Associativity occurs in a similar fashion as cooperativity, but an important distinction in associativity is that one of the pathways can make the postsynaptic partner fire. The terminology to describe this nuance is to refer to the pathway that can make the cell fire on its own as strong, and the pathway that cannot make the cell fire on its own as weak. Hence, in associativity, there is a strong and a weak pathway, but in cooperativity, all the pathways are weak. Nonetheless, the logic for associativity in terms of the NMDA receptor will be similar to cooperativity. The activity of the strong pathway allows to generate a sufficient amount of depolarization for the weak pathway to open its NMDA receptor and subsequently generate LTP there as well. Since we are on the topic, I'll briefly mention that these concepts such as cooperativity, associativity, and input specificity are key to understanding some fundamental aspects of memory, which, as I've mentioned, are mediated by the hippocampus. So, one way to think about cooperativity is that this property ensures that only the events that are really important will result in memory formation because there is a certain amount of threshold that they need to attain before the postsynaptic cell can fire. When it comes to associativity, if you're familiar with Pavlovian conditioning, you will recognize that the two concepts are similar to one another. A strong unconditioned stimulus, which is the strong pathway, will be associated with the conditioned stimulus or weak pathway to enhance the connection between the two. Lastly, input specificity ensures that only specific memories are encoded and retrieved. A final and important definition that I want to establish is the concept of homosynaptic plasticity and heterosynaptic plasticity. If we consider two different pathways interacting on a postsynaptic cell, when one says that LTP is homosynaptic, it means that it occurs at the synapse where the induction happened. On the other hand, heterosynaptic means that the change in response occurs at the synapse where induction did not happen. So, for example, in the associativity scenario, if we imagine that we only stimulate the strong pathway, from its perspective, there is going to be an X amount of homosynaptic plasticity. From the perspective of the weak pathway, there will be no heterosynaptic plasticity. Alright, with these definitions now taken care of, I want to address the aspect that LTP can occur through different mechanisms. To do so, let's consider the mossy fiber pathway between the granule neurons and CA3 neurons. Starting with the electrophysiological recordings, it turns out that applying a tetanus to this synapse will also induce LTP. However, if we perform the recording with AP5, you will notice that the LTP still occurs. It turns out that LTP in the mossy fiber pathway is entirely presynaptic, and for that reason, it does not require the activation of NMDA receptors. In this mechanism, sustained depolarizations by the tetanus cause large amounts of calcium to enter the presynaptic terminal. This calcium entry binds to calmodulin and activates a calcium-calmodulin-dependent adenylyl cyclase. Adenylyl cyclase converts ATPs to cyclic AMPs, which then go on to activate pKa. pKa then goes on to phosphorylate other proteins involved in the vesicle cycle, and that ends up increasing the transmitter release and the postsynaptic response. Now, if we do the electrophysiological experiments with a pKa inhibitor, you will see that the LTP now does not occur. Furthermore, due to the fact that the postsynaptic cell is not engaged, this form of LTP is said to be not associative. Alright, now that we have seen LTP in great detail, let's cover how long-term depression occurs. To see LTD, we can consider again the synapse between the CA3 neurons and the CA1 neurons. In comparison to LTP that was induced by a very high frequency stimulation, LTD arises from very low frequency stimulation of about 1 Hz for 10 to 15 minutes, and as you can see here, the responses after induction now become lower relative to the baseline. The mechanism to induce LTP is actually very similar to that of LTP because both are mediated by calcium entry from NMDA receptors. However, in LTD, because the frequency of stimulation is so low, small and slow rises of calcium lead the calcium to activate phosphatases instead of CaMK2. Remember that phosphatases are essentially the opposite of kinases, and their activity mediates the removal of phosphate groups. Two important phosphatases that have been pointed out as being activated in such contexts are PP1 and calcineurin. The activity of these two phosphatases eventually leads to the removal of AMPA receptors that get internalized back into the endosome. Just like in late LTP, there is also a late LTD that is mediated by protein synthesis. Now, to finish our discussion on plasticity, although the previous experiments that I have introduced in the hippocampus give us very good insights as to how plasticity is mediated, it turns out that the induction protocols are not very accurate as to what happens physiologically. Indeed, trains of spikes in the hundreds of hertz do not occur in the body, but yet plasticity does happen. A more physiological way to induce plasticity is spike timing dependent plasticity or STDP. In this paradigm, the induction is performed by pairing a presynaptic stimulus with the firing of an action potential in the postsynaptic cell at a low frequency. Instead of the frequency of firing, the main variable that we will focus on in STDP is the interval of time between the two stimuli. Generally speaking, when the presynaptic stimuli precedes the postsynaptic stimuli, it causes the connection to undergo LTP. On the other hand, if the presynaptic stimuli occurs after the postsynaptic stimuli, it leads to LTD. To illustrate this relation, we can consider a plot of the time interval between the two signals and the generated response. From this graph, you will notice that for LTP and LTD to occur, it is highly dependent on the time interval. Moreover, the time interval is very small, which means that plasticity can only get induced within a few tenths of milliseconds. When it comes to the mechanisms that underlie STDP, they are again believed to be linked to the NMDA receptors in a mechanism that is similar to what we have covered previously. One thing that I will note right now that will be important for us to understand these mechanisms is that when an action potential is triggered in a neuron, it turns out that it can actually backpropagate throughout the neuron back to the dendrites. So, if we consider the LTP scenario first, the presynaptic activity will cause the release of glutamate, and the backpropagating action potential will cause the required depolarization to kick out the magnesium and open the NMDA receptors. Hence, when the activity is pre before post, the NMDA receptors can fully open, let calcium enter the cell, and eventually lead to LTP. In the LTD scenario, because the glutamate arrives after the backpropagating depolarization, only a bit of calcium can enter the cell, which leads to a long-term depression by the same mechanisms we have discussed. Based on what we covered previously, you will notice that STDP follows the general principle of Hebb's postulate. If the presynaptic cell contributes to the firing of the postsynaptic cell, then the connection will be strengthened by LTP. However, the other side of this medal cannot be explained by Hebb's postulate. The formal extension to Hebb's postulate came from Gunther's tent, which basically said that the synaptic efficacy between the presynaptic and postsynaptic cell will be decreased by LTD if the presynaptic cell fails to contribute to the firing of the postsynaptic cell. In the context of neuronal development, we can interpret STDP as a simple mechanism that reinforces connections that make the postsynaptic cell fire and removes the connections that are irrelevant to its firing activity. Finally, you will notice that there is an asymmetry in the intervals of time that cause LTP and LTD responses in hippocampal cells. The interval for LTD is much longer than the interval for LTP, and this is most likely explained by the fact that since there is a lot of noise, the interval of LTD must be bigger to refine the connections and make sure that any unnecessary connections are removed. Alright, with the basics of plasticity now covered, we have essentially completed every point that I wanted to cover when it comes to the fundamentals of neurons. Now, in my opinion, the beauty behind the nervous system does not necessarily come from the neurons themselves, but rather from how they assemble together to form structures like the brain, the cerebellum, and many others. Accordingly, I want to switch gears in future videos and discuss topics like the anatomy of the nervous system as well as sensory and motor systems. Thank you for watching this video. If there was anything unclear or there was a mistake somewhere in the video, make sure to let me know in the comment section. If you enjoyed this video and found it useful, you can consider leaving a like and subscribing to support the channel. On the right, you will see the informational resources that I've used to produce this video. Thank you again for watching, and I'll see you in our next discussion.